In today's MedicoApps Masterclass, we will learn about antigen antibody reactions. Antigen antibody reactions are a specific, reversible, non-covalent biochemical reaction between antigen and antibody. If we see antigen and antibody, antibodies are smaller and always soluble, whereas antigen can be soluble or smaller or insoluble or larger antigens. If a soluble antigen combines with a soluble antibody, this reaction is called as precipitation reaction. Whereas when an insoluble antigen reacts with a soluble antibody, this reaction is called as agglutination reaction. Let's look at the various different types of antigen antibody reactions. Antigen antibody reactions can be in vivo or in vitro reactions. In vivo reactions happens inside the living organisms, and these are the reactions which leads to either elimination, destruction and removal of the antigens, whereas in vivo reactions are done in laboratories, and these reactions, by help of them, we are able to either detect the antibody or the antigen in vitro. Now, various in vivo reactions are agglutination reactions, precipitation reaction, complement fixation, neutralization, antibody dependent cell mediated toxicity and immobilization. Whereas various in vitro reactions are agglutination, precipitation, complement fixation, neutralization, ELISA, radioimmunoassay and immunochromatography, even fluorescent antigen antibody technique. So all these reactions, there are subtle differences, and essentially what happens is antigen antibody combines together in all these reactions. If we see the in vivo reactions, there are three stages. The primary stage, there is a formation of antigen antibody complex by weak intramolecular forces. So in the first primary state, there would be antigen antibody complex by weak intramolecular forces. These forces are van der Waals forces, ionic bonds and hydrogen bonds. So these bonds, the van der Waals ionic or hydrogen bonds, require the presence of sodium chloride and other electrolytes. So in absence of electrolytes, this antigen antibody complex formation will not occur. This is an important point to remember. Second stage, after the antigen antibody complex is formed, this leads to the precipitation, agglutination or even cell lysis. So this is the secondary stage. And finally, in tertiary stage, there is a destruction of the antigen or neutralization and even cell damage or tissue damage. So these are the three stages of antigen antibody reactions in vivo. Precipitation, as already told, when a soluble antigen combines with a soluble antibody, it leads to an insoluble antigen antibody complex. Now, sensitive in detection of antigen but less sensitive in detection of antibodies. Precipitation reactions are relatively more sensitive when it comes to detection of antigen, but they are less sensitive when it comes to detection of antibodies. Now, Marek in 1934 suggested a lattice hypothesis. What basically this hypothesis says that at correct concentration of antigen antibody, there is a lattice structure formation which leads to the precipitation. So when there is antibody excess, again the lattice will not form. When there is an antigen excess, the lattice will not form, but at equivalence ratio, the lattice structure will be formed, and hence the precipitation will be there. So this is the precipitation curves, which means that at this point, the point of equivalence, the lattice is formed. This can be seen in this experiment where this is the antibody antigen, and in a zone of equivalence or equivalence, the lattice formation happens. So this is the precipitation and the lattice after the lattice formation. Moving forward, let's look at some important examples of antigen antibody reactions. So this is the ring test already explained. Two examples of such kind of ring test is a streptococcal Lancefield grouping and Ascoli thermoprecipitant test. So these are ring tests, and again at the zone of equivalence, the precipitation happens, and you can see it in form of a ring. This is slide test in VDRL, as in syphilis. So this is non-reactive when there is no flocculation. This is weakly reactive, and when the clumps are clearly formed, this is a strongly reactive antigen antibody VDRL test in syphilis. Another example would be agglutination, as in tube test or Khan tube test in syphilis. Here you can see that the agglutination is starting at a titer of 1 by 40. One important brain teaser question would be, what is more sensitive, the agglutination or precipitation? Now, as already explained, that agglutination, the antigen is already large and it is insoluble. So very less amount of antibody can make the agglutination happens. That is the reason why agglutination is much more sensitive than precipitation. Again, this is a very frequently asked question, that which is more sensitive. So agglutination is more sensitive because in case of agglutination, the antigen is already large, antigen is already insoluble, so very less concentration of antibodies can also make the agglutination reaction happen, and so it is more sensitive. Moving forward, so another example of antigen antibody reaction in vitro would be immunodiffusion. In this, what happens, so this is a elk C. diphtheria toxin text, and what happens is, this is the patient isolate, which we don't know. This is the toxigenic C. diphtheria. This is non-toxigenic C. diphtheria, and these are the elk line. These are perpendicular line formed after the formation of the antigen antibody. So here the antigen will be coming from, here the antibody will be coming from, and then at perpendicular line they will be meeting. So this is an example of a positive elk test. Moving to countercurrent electroimmunophoresis, here what happens, the antibody is filled, this is an antibody filled through, this is the antigens, and when we pass on electricity, the antigen antibody moves toward each other, and at zone of equivalence they will form these lines. So we can detect multiple antigen antibody based on this countercurrent immunoelectrophoresis. Last example would be rocket immunophoresis. So these are the rockets of antibodies. Here what happens, the antibody is in gel, and then we put antigen in form of these platings, and this is a qualitative method in which what happens, the size of the rocket actually determines how much that antigen is there. This is a qualitative method, and the length of the cone indicates the concentration of the antigen. Closing this topic with the most important points of antigen antibody reactions. So precipitation is soluble antibody with soluble or small antigen leading to insoluble precipitant. Agglutination is soluble antibody with insoluble or large antigen leading to agglutination. Agglutination is more sensitive than precipitation. Antigen antibody reactions cannot occur in absence of electrolyte. VDRN is an example of slide flocculation test, and rocket immunophoresis is a quantitative test. Thank you.